All right, thank you for having us. Kristen and I work closely together, so we combined our presentations, and I will try and get it shared now. Hopefully you can all see that now. All right, as we get started, I just wanted to give a couple definitions. We use a lot of acronyms, as do other groups, and I have a tendency to use them without defining them, so I just wanted to go over them really fast. The BioMer is the Biospecimen Procurement and Epidemiology resource, and it is a new shared resource with this last submission for the P30 grant for the Cancer Center. And what it is it is combining Tissue Procurement Core, the Molecular Epidemiology resources, or MERS, and Orion and Perch, which all work together, and I'll give you quick overviews of both of those as we move through this. Perch I will talk about in just a second, but it's essentially the unified biorepository consent that the Cancer Center is using to enroll patients to observational and longitudinal projects. Kristen will give you more information about the Tissue Procurement Group. Orion is the Oncology Research Information Exchange Network, and that's an alliance of 19 cancer centers that we work with collaboratively to enroll patients to follow long-term, but also send in tumor material and germline material for molecular sequencing, and we get that raw data back for investigators to use locally. And then the MERS will definitely go into more detail. This is basically just a picture of what the BioMer is hoping to do by bringing together those three groups. We're really trying to work together more cohesively as a unit to maintain prospective observational registries that includes the MER and Orion, to collect and process quality biospecimens for both clinical and translational research, which is Kristen's team, and then to provide more of a centralized resource for investigators. Historically, they've had to go a few different places when they wanted access to tissue or other materials, and we're trying to make that easier on everybody. This is really just a figure showing you what the unified consent looks like. When we joined Orion a few years ago, it became apparent that we were going to be approaching many more patients for consent. And the previous way we were doing that, with each MER or disease repository having their own consent, was going to mean that we were approaching patients with multiple consents and asking them to provide multiple signatures when we're really asking similar permissions across the board. So what we did is we created Perch, or the Unified Biorepository Consent, and that includes the 8 MERS, or disease-specific resources, the gynecology group, and then the Tissue Procurement Core. With that consent, we are able to approach all new patients to participate. Depending on staffing levels and eligibility, we're approaching mostly patients that are going to surgery so that we can collect specimens. And as we continue to grow, we're hoping we have e-consent functionality, but hoping to improve upon that so that we can approach patients, more patients than we're able to do in person right now. Let's see, if they're eligible for a MER, which is the disease-specific repositories, they do get approached with the Perch consent, and then what is an addendum, which is essentially just an abbreviated version of a consent. So they do provide 2 signatures, but they're presented frequently at the same time to try and decrease confusion. And the permissions include collection of all types of specimens, long-term clinical data, many patient-reported outcomes questionnaires and quality of life questionnaires, and essentially permission to contact in the future for other research studies down the road. This is the Perch enrollment to date. About half of these participants are also enrolled in MERS. So right now we're getting close to having 5,000 patients that we've consented since we opened, and enrolling in about 60 to 70% of the cancer center clinics, and working with both the lung and the head and neck group to try and build a better population in that group. Our challenges to date have been with head and neck, identifying the patients early, potentially before they come to the cancer center, and trying to find space to enroll them in the head and neck clinics. And then lung has been identifying the best ways to get material and to enroll in those clinics. So the MERS, which is what I'm supposed to be talking about. The molecular epidemiology resources began in essentially 2002 with the lymphoma group. And what they are is they're disease-specific registries where if they have a certain type of cancer, they're asked to participate, and the patients are asked to provide samples, allow us to follow their medical record, whether they're being seen locally at UIHC or closer to home. We can ask for those outside records. And then also send questionnaires for patient-reported outcomes or quality of life. This is enrollment over the years. I combined basically 2001 to 2007, which is when we were primarily focused on lymphoma, and then started to open additional MERS. So you can see kind of the timeline below. Started recruiting just in lymphoma, and then melanoma and sarcoma were combined into one project in 2008. And then the breast team wanted to follow suit and opened a similar project in 2010. Sarcoma and melanoma split into two separate projects in 2015, so that's why that is there. And then subsequently we've opened MERS in GI, GU, myeloma, leukemia. And then the gynecology group isn't technically a MER, but they collect specimens and are able to go into the record to collect clinical data when projects come up or they're asked to. Then the UBC is PERCH. So you can see this big jump is when we opened PERCH and started enrolling patients to both of those. This is an example of some of the specimens that are collected. So each MER collects similar specimens, but not always in the same way. Some will do serial, some will not. But most will collect a baseline serum, a baseline plasma, some buffy coat, and the majority are collecting a baseline blood DNA. For Orion, we have been collecting buccal samples. So if we don't have the opportunity to collect blood for the germline DNA, we can get those cheek swabs. And then the PBMCs and the mononuclear cells are more recent. Well, I guess both old and more recent. We used to collect a lot of them for lymphoma, and we're starting to get into that again. But then with the myeloma team and the leukemia team, we're starting to do more of that. And then as far as tissue specimens, Kristen will be able to speak better to this, but for the MERS, we try to collect tumor for patients that have been enrolled and meet the eligibility criteria. And then if they're also eligible for Orion or PERCH, we try and collect adjacent normal in the event that we're not able to get that germline material from blood or from the buccal cells, so that we don't have to hold back on shipping the specimens in. And I can share these slides for numbers and that with people later. These slides in Paraffin are really specific projects where we hold archival specimens and shared either slides or cores with, or scrolls with researchers for collaborations. But we don't currently fix what we collect. It's more fresh collections for distribution, or they freeze it. And I will turn it over to Kristen, who has a lot of information about the Tissue Procurement Core. And I'm going to continue to drive, so she's going to tell me when to advance.
Okay, hello everybody. So I am now currently the technical director of the Tissue Procurement Core. Our medical director is Dr. Mike Knudsen out of pathology. Our mandate is with both the Carver College of Medicine and the Holden Comprehensive Cancer Center to support all research projects, both cancer and non-cancer. We do this by collecting tissue from a variety of sources, predominantly out of the OR right now. So if a project comes to us and say you want some gallbladders, like some healthy gallbladders or some as close to healthy as you can get that are being removed, you know, we would go and we would consent your patient to the UIBB, which I'll talk about in a second, the University of Iowa Biobank. And then we would put in an order in collaboration with surgical pathology. And if the tissue comes out of the OR and there's enough that they feel comfortable distributing for research, we would then collect that tissue, provide it fresh in media, or we could freeze it down, or we can fix it. And just whatever the project needs. We're kind of developed into a boutique service, so we have researchers coming with a variety of different project needs, and we try to meet them. As Laura gave a little bit of background on this slide, prior to 2019, we were mainly College of Medicine, but we were mostly doing oncology research. So the Cancer Center kind of stepped in and is giving us some more functional oversight now. So we kind of work for both teams at this point, cancer and non-cancer. If you can advance to the next slide, please. The University of Iowa Biobank is our banking resource under the Tissue Procurement Corps. It's housed in the Medical Research Center. We have a extensive freezer farm down there. As I mentioned previously, the majority of our current work is prospective custom collections designed to meet investigators' needs specifically. But we do have a fairly large repository of fresh frozen tissues, which are separate from the MERS that Laura just showed. We tried to focus on tissue types that the MERS were not collecting to provide University of Iowa researchers a more well-rounded biobank if you look at it in total. Our tissue is processed in OCT and stored in liquid nitrogen vapor in a cryosafe. The little image on the side just shows since I took over in the beginning of 2019, we've moved even further from banked requests towards custom collections. Next slide, please. So just a little bit about how we cryopreserve. We consent the patient, we order the tissue as I mentioned, and then once we have the tissue on hand, if we're freezing it, we use the snap frost, which is isopentane immersion method. It's currently the gold standard from ISBR, so that's what we use rather than liquid nitrogen vapor freezing. And then we store it in the liquid nitrogen vapor in the cryosafe in advance. This is just a picture of our cryosafe. It's a pretty spiffy machine that I inherited. So we have quite a bit of room to store all of our tissues, which is a little bit better than keeping them at -80 or even -150. If you could advance. So I've included some selected bank tissue numbers. Again, this is mainly focusing on things that the MERS weren't collecting. For a long time, the MERS weren't collecting colon, so we collected quite a bit of colon. The MERS do collect kidney, but we have a large amount of kidney. Mainly, we focused on lung, which, as Laura mentioned, hadn't been really, collected under the purge consent up until now. So we do have all of these tissues. The majority of them, I've included primary, malignant, and adjacent. The majority of them, we do have adjacent tissue on the same patient. And how that is determined, whether it's adjacent or not, is just grossly. So the majority of our tissues do have to be QA'd. We're not prospectively QAing until we have a project that comes in. So I've included just, a little snapshot of a couple of projects that we've done. Under the Tissue Procurement Corps, we also have what people sometimes refer to as like the DNA Extraction Corps. It's different from the Genomics Corps under Kevin Knudsen, but basically we're just, isolating DNA from a variety of fluids. And we work with different researchers to obtain blood and other fluids as well. Like we have a couple of projects that are looking at cerebrospinal fluid. We're going to the place of collection and then spinning it down for them, aliquoting it out and freezing it. We do a lot of those types of projects, pretty simple stuff. For this project, we worked with some researchers here, including Pat Winokur, to go to micro and to identify some patients from microbiology and obtain blood samples from chemistry that were left over that were drawn around the same time as the blood culture. And we were able to provide those for testing. You advance on this, obviously, like, went on to become published. So we were happy to be able to provide these samples that otherwise would have been fairly difficult to obtain. Next slide. An example of a project with tumor tissue, working with Dr. Burnett and her team. We were able to provide tumor and matched normal tissue slides for RNA isolation. So we cut the slides and we're able to provide those to the team. We could do also IHC on frozen or FFP. We do that in partnership with the Department of Pathology and Dr. Belize and everyone over there that's working on IHC. Next slide. This is just an excerpt from their paper where they were looking at, epidermal growth factor and head and neck cancer cells. So we were able to go and consent and collect these head and neck carcinomas. Next slide. We then also worked with Dr. Allen in radiation oncology to prospectively collect non-small cell lung cancer and adjacent non-tumor tissue. They wanted this cryopreserved in OCT and then sections cut for staining. We've done that quite a bit. We've cut a lot of sections for the Orion project as well. So we are pretty well versed in frozen sectioning. And then they were able to go on and get a nice publication out of this, looking at oxidative stress. Next slide. This is a little excerpt from their publication. Okay, next slide. And then finally, another project utilizing tumor and normal tissue. We were able to come up with a way, and one of the harder things that we've had to do is establish a workflow for obtaining skin. A lot of researchers have come and wanted to obtain and utilize skin, including Dr. Saul's group and Dr. Quingelhutz and Dr. Mowry as well for their viral studies. So we have established with surgical pathology a way to collect this relatively normal skin from breast reduction surgeries. We consent the patients, again, place the order, and then collect and put in media. We work in conjunction with MAST, which is one of the MERS, to obtain melanoma cells. So they were able to compare the cancer cells with the relatively benign, fresh, normal skin cells. Next slide. And then an excerpt from their paper, which is really interesting on the coalescence of the melanoma, but not the normal melanocytes in there. Okay, next slide. And then finally, just an overview of our services. As I mentioned, the majority of our projects are just for procurement. People come to us. They want to obtain certain tissue type, whether it's oncology or non-oncology. We work on consenting the patient to either our protocol or one of the MER protocols to make sure that the patient understands, the whole process of what they're allowing their tissue to take part in. And then, we can process the tissue however they need. However, we also have moved into peripheral blood mononuclear cell isolation. We're doing a lot of work with the Cancer Center for a lot of their clinical trials. However, we're starting to have researchers come to us asking for PBMC isolation in a translational nature. So that's also available to investigators if you are interested. Something that I didn't list on here is decirculating tumor cells. Dr. Pashtun Kasi has obtained a machine that enables mechanical sorting of circulating tumor cells. So we also have, with collaboration from his lab, that capability. We specialize in RNA and DNA extraction from blood and from tissue and saliva. From the tissue, we can extract RNA and DNA from the fresh or the frozen, and right now we're offering DNA extraction from FFPE as well. And we actually have a new project coming in with Dr. O'Rourke to extract DNA from like 350 FFP blocks. So that is going to be exciting. As I mentioned before, CSF and blood processing and aliquoting will go to where the blood and the CSF is being obtained. We'll process that and aliquot that per protocol. We'll do frozen sectioning. We work with Dr. Kevin Knutson on some projects to do some of the PCR that is necessary to do some of the fluorescence assays that he runs in his core. And then we offer extensive choices on biospecimen storage as well. And that is it for me. I'll turn it back to Laura for the project process.
As you were talking, Kristen, I realized that I missed a slide and should have included that we started tracking all of the specimens that we collect for research, at least through the Cancer Center, in Lab Matrix, or also known as UI BioShare. So it's all in one laboratory information management system, so we're able to query across groups. So if you were to approach either the Tissue Procurement Core or one of the MERS for a project, and the MERS had a patient consented and had the clinical data, but didn't perhaps have the tumor tissue, but the Tissue Procurement Core did, we would be able to query that easily using Lab Matrix or UI BioShare to do that. Those are really synonymous. Lab Matrix is the software, and UI BioShare is the local branding of the service. In order to request access to a project, either with the Tissue Procurement Group or the MERS, we have initiated a new kind of single stop process, which is driven through REDCap survey. We are actively updating our representation on the website for the Cancer Center, and we'll have the link live there. But until that is live, you can reach out to either Kristen or myself, and we can send the survey to you. But essentially, there is a short form that comes out to investigators to really just give us an outline of what they're looking for. And then that will get sent back to the BioMer research team, and we'll triage it to the appropriate group, depending on what population you're looking for or what sort of information you're trying for, specimens you're trying to access. From there, if it has not already been discussed at the MOG level, as far as utilizing specimens, it does go to that group, the MOGs or the MERS, just to talk about utilization of those banked specimens to make sure that it's an appropriate use of the banked cases. If something has already been through a grant review, then that kind of scientific part is skipped since it's already been reviewed. But feasibility, as far as how quickly can we obtain these materials, do we already have them, that kind of thing does get talked about as a group. From there, it goes to BREC, which is a newer committee, in parallel to TREC, which is the Clinical Trials Resource Group, which is really just a stop with Kristen and myself and Dr. Chan, who's the medical director for the BioMer, to look at our resources to see if we have the staffing we need in order to meet the request. If we don't, then we work with the investigator to figure out how we can accomplish what is being asked. Within REDCap, there are several other forms that are managed by our research team, which will include kind of an agreement that has an outline of what the scope of the project is, what the deliverables are, if there's a budget associated, so that everybody's on the same page and knows what's expected at the end. And then it eventually gets assigned to a BioMer staff member who can help process things through the IRB if an IRB is required, or through sponsored programs if there's contracting required at any level, specimen sharing, that kind of thing. And then annually, I think we're going to submit another survey that will go out to investigators that have utilized the resource, asking for updates on publications and grants so that we're able to track that a little more closely. that's been fairly manual to date. So we're trying to get a little more efficient with that process. But that is really the end. We didn't go over much with the medical director, but Dr. Chan is a GI surgeon and has been the director for the MERS for a few years and is now taken over as medical director for the BioMer as well. I'm the administrator for the BioMer, and then Kristen, as she mentioned, is the technical director, and any of us can answer questions that anyone has.
Okay. Thank you for a wonderful presentation. What a valuable resource. I'm almost afraid that other people know about it because you may be overwhelmed. What do you do for cost to fund all these activities that you do? And if a new investigator is interested in using some of these resources, what's the process for funding that's required on their end?
It's a good question, Kristen. You can probably speak better to kind of the pilot projects. Something that we're trying to begin doing is basically diversify the portfolio so that we have some maybe pharmacy supported or pharma supported registries that are going to help support the pilot projects and those projects that aren't grant funded, because we're fully aware that a lot of our resources are being asked for applying for grants or that kind of thing. So we're asking in those applications, maybe there's a % effort assigned to the coordinator depending on what the project is, or support from the Cancer Center through seed grants. It just really depends on kind of the situation. Kristen can speak more to how the TPC is managing it.
Yeah, I mean, the TPC is trying to focus on being able to supply tissues to newer researchers. I mean, that's kind of a passion of both Mike and myself. So if we're able to piggyback collection on one of those industry sponsored projects, we're able to do at cut rate for new investigators. And that's something that we're trying to do. That's why we maintain some of these industry contacts to try to provide tissues in a way that we can subsidize internal investigator research. That being said, it's kind of it's a case-by-case basis. If there's no, if the science is there and, you know, the effort is there in terms of, you know, time and things like that, then on a case-by-case basis, we review it at the BRAC and try to come up with a way to supplement the cost of collection so that we don't turn any research away.
We're also trying to be a little more transparent with our price list. A couple months ago, we worked with the CRU to look at kind of, you know, broader market values and what different institutions are charging for use of these resources, and have created a price list that has, you know, the base option, and then either the F&A increase if working outside of the institution, or Cancer Center members or College of Medicine members have a decrease from that base to try and make it more affordable, while also providing some support for our resources.
Thank you for that information. Any other questions from others?
Yeah, I have a question. If one needs to have, say, epithelial tissue from the lung or airways, can one specify that they want it from a donor/cadaver that was a non-smoking individual?
Yes, we can obtain that information. If we're looking at it retrospectively, like if it's from banked tissue, we have full access to the medical record. If it's from prospective, we can certainly ask that question when we go approach the patient for consent to determine their smoking history. We have one NCI project that is really picky about like exposure history. So we have a whole like pre-screening questionnaire that we have to ask the patient. So if we're doing prospective collection, we can certainly administer a screening questionnaire.
So in the case I'm thinking of, when we got tissues, the 2 donors had been killed in motor vehicle accidents and turned out were both smokers. And so the tissues raised that issue. Well, is this a good model for what you were trying to do? So going forward, we'd want to be able to request that the cells came from donors who were non-smokers. And I just wondered about that as a possibility. So these were fresh cultured cells from lung epithelium.
Yep, we can definitely screen by smoking. Since we mainly don't deal with autopsy, that's Dr. Nishalski's territory, we're mainly dealing with the patient in front of us. We're able to ask all those questions to make sure that we get appropriate tissue freshly for our researchers.
Okay. Thank you, Kristen.
Yep.
Any other questions?
This is Chuck. I guess for Laura and Kristen, how do you relate to the research histopathology lab in the Department of Pathology, or sometimes known as comparative histopathology lab?
I'm gonna let Kristen take that as well, because she is our primary bridge between pathology and our team.
Yeah, we work with Mariah down there quite a bit. Most of what we do with the MERS, if it's not associated with a clinical trial, if we need slides cut from FFPE blocks from the pathology archives, for instance, for one of our consented patients, we'll work with their team there. But as far as the fresh collections, most of that we're handling in-house. It's just the FFPE stuff. We'll work with Mariah.
So Mariah has sort of a charging mechanism for pretty much anything they do in that lab, including just mounting block so that they could cut it on the cryostat. Do you have a similar charging mechanism in the tissue core?
Yeah, Laura has when Laura took over administratively, when we moved to the cancer center, we tried to bring our pricing more in line with both prices from the CRU, prices that Mariah is charging, pathology prices, so that we're all kind of charging the same for the same type of work. So Laura does have a pricing list that she maintains that tries to approximate costs across the UIHC.
And in that regard, when you're dealing with a cancer center member, do you give a discount to them for the cost, for the cost of processing tissue?
We do. We give a discount to both cancer center members as well as the Carver College of Medicine investigators, since we're supported by both.
And where does the funding come from for that cost reduction?
So that's where we try and reach out to pharma partners or NCI projects that do have budgets associated with them that can help fund some of these projects that may or may not have that baseline funding available. So if it's a pilot project or a new investigator, we work to identify, I guess, we participate in those observational registries so that we can support the other projects from the revenue that we get from those.
And if you don't have revenue, do you end up sort of holding up a study because it can't move forward because of lack of funding?
We try not to. That is part of the reason that we established BREC, so that we can work with Kristen and Dr. Chan to identify what the requested resources are and if we can meet those needs, and if we can't, work with the investigator to identify what needs to happen in order to be able to move forward with the project.
Yeah, there are other internal funding mechanisms. Dr. Weiner is pretty supportive of the MERS as a whole, so we've definitely utilized those other mechanisms where a very well thought through project comes through. It requires a lot of resources, something that we can't maintain. Then we presented to Dr. Weiner and the clinical trials group as well to see if there's some way to partner with them. But we have a couple of NCI projects right now that we're working to utilize, and Laura has the registries with the MERS as well. But our NCI projects are funding most of our efforts so that we can provide these resources to internal investigators at a cut rate or sometimes even just for free.
Thank you.
Any other
I have
questions?
one question. Yeah, this is Patrick. Thank you, Laura and Kristen. As the director of a facility within the center here, might get investigators wondering about this resource. Could you just kind of step through the process, or what I would tell them to? Is it just a matter of getting a hold of you, Laura, first, or what would you recommend there?
Sorry, I was muted. You could reach out to either Kristen or myself, and we can get you pointed in the right direction. Otherwise, once we have the Cancer Center website updated, which is in process, so I'm hoping it'll be live in the next week or so, there will be that link to that survey so that you could initiate into the kind of queue that way. But if you were to reach out to Kristen or myself, we can also send that survey and make sure that we have whomever it is on the list. And if it's just the survey is really just, what are you looking for? You don't have to know specific details. So if it's somebody that just is asking, I know I want to collect tissue from a certain site, but I'm not exactly sure what I need, that's all that we need in that initial survey. And then Kristen or whomever will follow up to get more details.